Okay, everybody, let's get started. I would like to welcome you to this beautiful sunny afternoon here in Berlin. And I would like to introduce myself first. My name is Dietrich. I am the Global Product Manager for our DNA extraction products, meaning DNA extraction chemistry, as well as DNA extraction instrumentation. And I would like to give you a warm welcome from my side. Thank you very much for joining this webinar. And in the next 30 minutes, I will present a little bit about our new DNA extraction kit for livestock samples called sbeadex Livestock Kit. And I would like to give you a brief introduction about the benefits and the specifications of this kit, show you some kind of application data, and provide you with all other relevant information about this new kit. So thanks for joining again, and I hope that we'll have a good time. So if you have any questions, please, I will show you my internet address and my email address after this webinar, or you can simply use the chat function. I will have a look if I can answer this question immediately. So, livestock, always an interesting thing, a very interesting topic. We see right now that especially genomics are getting more and more involved in this topic, and that there's a lot of activity right now in this topic, and therefore we have thought to think about our livestock strategy at LGC again. So here you can see our DNA extraction portfolio for livestock samples before we have launched this new kit, meaning that we have a couple of different kind of sbeadex kits, for example, sbeadex blood, sbeadex tissue, sbeadex forensic, sbeadex plant. And these kits were more or less specialized on different kind of livestock sample types, meaning, for example, for blood card, we had the blood kit for sure. The tissue kit was designed for tissue samples, like ear punch samples from different kind of animal species. Also, the sperm and semen samples were extracted using the sbeadex blood kit. For hair samples, there was a need to use a sbeadex forensic kit because hair samples might be very complicated and the DNA yields are quite low sometimes. And for plant samples, for sure, we had our sbeadex plant kit. And that was the situation there, meaning different kind of kits for different kind of plant and animal species. And this means also that there were different kind of protocols for different kind of sample types, meaning that mixed sample runs, for example, 10 blood samples together with 20 tissue samples, together with 10 hair samples on one run were not possible at this time. So different sample types and specific kits were required. And we've thought, okay, it might be useful for our customers, for our service labs, but also for our internal services to make this a little bit easier, meaning that it might be really a big benefit if you can use just one protocol for all different kind of sample types. And that's exactly what we have done. So first of all, again, a short summary about the current situation. So the lysis is done offline. The lysis buffers are sample specific. I've shown you that. The protocols are also sample specific, so sometimes you might need to wash longer than for other samples. Also, the elutions were different, lysis conditions were different. That's all really sample specific. Mixed samples per runs were not possible at this time, meaning that you have to extract your hair samples first, then the tissue sample, then, for example, the blood samples, and you can't extract in parallel. The DNA quality and quantities were always high. Our sbeadex kits are really providing you with high DNA quality, and you can use this DNA for different kind of applications, including next generation sequencing, genotyping, PCR technologies and arrays, and manual and automated extractions are both possible. But again, we have thought about making it a little bit easier for our customers, meaning that you can just use one protocol for all sample types. And what does it mean? In the end, you still have different kind of livestock sample types like tissue, blood, plant, hair, or sperm, semen samples, but you all can extract these samples by using just one kit. and just one protocol, which is then called the sbeadex livestock kit. So the sbeadex livestock kit has the possibility to being used for all different kind of sample types. So one protocol for all livestock samples, and that was the basic idea to make the life of our customers a little bit easier. If you now have a look in a comparison, compare our old portfolio, the old sbeadex kits with the new kits, you can see the two differences here in this table. So in the past, we were using multiple different kind of protocols for different kind of sample types. Now we're just using one protocol for all different kind of sample types. And therefore, it is possible to have mixed sample runs, meaning that you can extract, for example, 10 blood samples, 10 tissue samples, and 20 hair samples in parallel on the machine. So all the other things are still the same, meaning that you still have to do the lysis often of your system, that you still have to use different kind of lysis buffers and different kind of lysis conditions before you're starting the run. That means also that the DNA quality and quantity is still very high, so you still can use the DNA for all kind of downstream applications like sequencing, like genotyping, PCR arrays, and so on. And for sure, you can use the DNA livestock or sbeadex livestock kit manually, as well as on all open platforms like Kingfishers, like Tekens, like Hamiltons. And if you have any question there, we for sure provide you with our support from our application team. So, going a little bit more into detail, what does it mean? You might be aware of this if you know already our sbeadex technology, but I would like to show you some benefits and differences of our sbeadex technology in comparison to others. First of all, again, the lysis buffers and the lysis conditions are still sample specific, meaning for tissue you're needing a special lysis buffer SB. And if you compare this lysis buffer SB, for example, here for tissue, with the blood one for FDA cards, you can see already that we're here using slightly different lysis conditions and lysis buffers. All these kind of lysis buffers are in the kit anyway, so you don't have to buy anything extra. If you are looking forward to buy the kit, you will see different kind of lysis buffers, so that you can also play around a little bit if you want, or if our application team gives you some recommendation in terms of lysis conditions, then you can simply follow these recommendations and you don't have to buy anything extra here. So what you can see also here, that for hair samples, which are quite common, especially for bovine samples, we are still recommending to use our hair lysis buffer. The reason is simply the nature of the sample, because a tissue sample, for example, is totally different to a hair sample, because here we are just having the follicles which contain the DNA, and for tissue samples you definitely need protease to digest the tissue overnight. So again, if you have any questions here, please let me know. We are very experienced with all kinds of sample types, and there might be also other things which might help you to increase DNA yields and which gives you the right support. For example, also in semen cells, and you have to shear your sample sometimes so that you can elute the DNA and so on and so on. So please let us know if you have any questions with our sbeadex technology or in general. But generally then we are following our normal sbeadex technology workflow, meaning that we're starting the lysis. I've just shown you this, but going on then with first binding step. So all of our sbeadex beads are coated with a silica matrix, and the DNA, which is shown here as this noodle-like structures, this is bound then to this silica coating, which you can see here around the metal core, which is this blue circle. And then we are starting with the first wash step, where we're using salt-based buffers and alcohol-based buffers. But then you can see here the difference between our extraction technology and other suppliers. We have a second binding step here, and you see already this Y-like structures here, and these are the so-called adapters, meaning we are really switching our binding technology from a polarity-based binding during the first binding step to a more or less adapter-specific binding, which we're using here during the second binding step. And the big benefit here of this second binding step is that we can simply use other washing conditions, meaning we are using pure water for the final wash step here. So we don't use any ethanol-based buffers anymore. We are simply using 70%. We are not using 70% ethanol. We are using water. And therefore, you can remove all kind of contaminations or things which might inhibit the downstream application very efficiently, so that we can really achieve high yields and also high purities. The second big benefit of this pure water wash step is that you don't need to evaporate your ethanol anymore. So normally, if you're washing with 70% ethanol, you have to wait for 10, 15, 20 minutes till you have a dry pellet again. But as we're using pure water, this kind of evaporation step is not necessary, and so you have a time-saving effect here as well. So normally, this kind of sbeadex technology is a very quick technology and also very flexible in terms of these kind of things. So please keep that in mind. A general summary about the general benefits of sbeadex. Again, we can use this kind of technology for all open platforms, including the Kingfisher Flex, for example. But for sure, we have optimized it for our own platform, which is called the oktopure. I will come to this a little bit later. What is really important to know is that sbeadex is very, very flexible, meaning that you can play around with different kind of elution buffers, wash buffers, lysis buffers, and this kind of flexibility, for example, would also allow you to design the protocol according to your needs and requirements. Just an example, if you would like to have a quicker protocol, for example, to increase your throughput per day, this is definitely possible. We can play around with washing conditions and also incubation times, but please keep in mind that this might have an impact on your DNA quality, meaning that you can use the DNA maybe still for PCR-based technologies, but if you're looking for next generation sequencing, that's something what you have to test if this is possible or not. But in general, we are often designing these kits according to the requirements of the customers, and we can really play around with different kind of conditions here and also buffers. Again, you can use the DNA normally for all applications. It is one of the big benefits of magnetic beads in general, that there's a certain grade of scalability. So here we are saying up to 2 ml. We always have to double check that if your robotic equipment is also doing a good job here, meaning, for example, mixing the beads and so on and so on. So that is also something which is really depending on your robotic equipment in your lab. And you can really adapt this to all matrices. So we are not limited to livestock plant samples. You can also go for plasmid and typical forensic samples. So that is a short summary of sbeadex. And I would like to continue now with the first look on the species and sample types which we have tested. And this is really a growing list, and it's really, there are new animals coming up, not from day to day, but from week to week. And but I've simply listed, I think, the most important ones which we have met so far in our labs and where we have experiences with as well. So I think bovine is really quite common. Pork samples are really also everywhere. Chicken is a very important one, where we have tested feathers as well as blood, for example, and also tissue. Goat samples, sheep samples, normally like bovine, a lot of ear punches here, but also hair samples sometimes. Horse, also very important, not only in the Arabian world, but also in Europe and North America. But you can also use this kind of kit for insects. So a lot of producers and suppliers of insecticides who are really killing their animals are using the kit. You can, for example, try to extract DNA of legs as well as the abdomen, so the body of the insect, and have a look for example, mutations. So the livestock kit can also be applied not only for real livestock, but also for all other animals which you know. Fish breeding is a quite hot topic at the moment because the oceans are empty and there are not so many fishes anymore which can be eaten by the people. So fish breeding is really a hot thing right now, and we're working together with some big fish breeders as well there. And for sure, what I've mentioned, it is especially for service labs quite important that you can use this kit also for all kind of plant samples. So there's again a long list of different kind of plant species and sample types what we have tested, and you can use this livestock kit and the protocol also for plant samples as well. Sample types, again, very difficult sometimes to extract because the nature of the samples, I will come to this with one of the next slides. Hair samples are quite difficult sometimes, especially if there is no follicle. So the DNA yields might be very low here. Ear punches are always depending also on the sampling process. For example, how these ear punches are stored and how old they are. Other tissues like, for example, livers or skin or whatever you have are also possible. For sure, you can use the kit for frozen and fresh blood, which are stored in FDA cards. Semen and sperm samples are also possible, no problem at all. Again, these kind of sample materials sometimes might need shearing or any other treatment, but for this kind of things, please contact also our application team because they will be able to provide you with the right information, the right protocol right from the beginning. And for sure, plant leaves and seeds are possible as well. So we are very experienced with all kind of plant types, sample types. Also, what is getting more and more popular is because it is nothing painful for the animal, are swabs and saliva, but this is also no problem at all for the kid. Now I would like to give you some examples in terms of results and applications, and I would like to start with some blood samples. So what you can here see is that the normal input is about 100 microliters, and the elution volume is about 80 microliters. I will send you the data anyway after this webinar, and so that you don't have to write down these numbers now, and that you have an idea about our typical concentrations and also our UV measurements, what we are also doing routinely in the lab. So what you can see here is already that we have experiences with different kind of blood samples, and what you can see here also is that the only exception where you need a dilution is chicken or avian blood, because here, as you might know, all blood cells are nucleated, meaning that you also have, for example, DNA in the red blood cells, for example, and therefore you cannot start with 100 microliters of pure blood. You have to dilute this blood 1 to 50 in, for example, PBS buffer. But in the end, the concentration and also the UV measurements looks fine, I think. What is important also is if these kind of concentrations are too low, or if maybe the total DNA yields are also too low, you can simply scale this up. Again, we are talking about 100 microliters starting volume of blood. If you're needing higher concentrations, then you can simply reduce the elution volume, or if you're needing more DNA, then you simply can also increase the starting volume. That is again something which is possible, and I think that's something you can discuss also with the application team. For sure, we have also tested some tissues here, and tissues might also be FDA cards. Here we have tested also not only fish but also bovine, which is stored by this typical all-flex sampling systems. For sure, you can also use cases, for example, or other systems which are quite common in this livestock market, especially for tissue samples. And what I would like to mention here is also that feathers are possible. Again, I really recommend in terms of these kind of applications to talk with our application team, because you have to be aware of that there might be, for example, also bacterial DNA in your samples, and that you are getting not pure bird or chicken DNA here, but maybe also other things. So please, that is something you should always keep in mind, and especially if you're talking about avian sample types. But generally, again, we can design the protocol according to needs and ideas, and this is also relevant for all tissues. Finally, I would like to show you some data for sperm swabs and plant. So swabs are saliva samples. For sperm samples, here we have simply data for pig sperm and bovine sperm. For sure, we have tested also other sperm samples already. For swabs, this might be, for example, from dog or pig or whatever. And finally, last but not least, plant samples are always important. For plant samples, again, there's a long list of about 80 to 100 plant samples which we have tested. And this kit will be able to extract of about 90% of all sample types. There are some special candidates which might need a special treatment in terms of reducing agents or other things. But in general, if you have the most common plants in your lab, then that is no problem for the kit at all. Again, because I think it's important, livestock samples are very heterogeneous, meaning that the nature of the sample is very important for the performance of the kit, and also the sampling process is very important for the kit. So these kind of application data, what I've shown you, might be good for our samples which we had in our lab, but for all other samples it might be a little bit different. The reason is simply that samples are stored differently, so storage conditions are often very important. For example, if you're using an all-flex sampling system or a cases sampling system, also in terms of the FDA cards, often the age might be important of the sample. So how long are the FDA cards already stored somewhere, and in which temperature, under which conditions? And for example, also as hairs are still quite popular in terms of sampling of livestock samples, it is really important that you have the follicles of the hair also in the sample type, because most of the DNA is coming from the follicle and not from the hair itself. Meaning, if you have no follicles, then you have really low DNA yields in total. So that is really important. And so please keep in mind that the sampling process is really important, influencing your final DNA extraction performance. And also the nature of the sample is important. Just an example, we had to realize that the age of the animals is really important as well, because there are simply differences between tissues of young and old animals. This might be that older animals have a thicker fat layer in their skin. It might be that they have a higher level of hairs in the skin. It might also be that there are other things which are important, which I really don't know now. Please keep in mind that age of the animals might be really something which is really influencing and having an impact on the final DNA extraction. And also we are seeing sometimes territory and environmental impacts. So sometimes there's really difference if you have animals which are free and living on the green grass outside, or if you have simply animals who are in the farm all the day. So that there's really sometimes interesting differences in terms of the nature of the sample, and so these kind of things might have an impact as well. Generally, as it is quite hard to predict these kind of things, so this kind of extraction specifications like DNA yields and quality and so on, we are offering pilot studies, meaning that you are always allowed to send and ship us sample material, which we then extract here in our application labs, and which then can be tested with the announcement application. So we are always offering this kind of application support, because we would like to be sure that our kit is covering all kind of different kind of natures, and also really can be used for all kind of animal sample what you can imagine. So we are always interested in working together with you, and this is, I think, a great thing. So you are getting the full scientific and application support from us as well. If you have any questions here, please let me know. I will also show you my email address in the end, and you can write me an email. Really, our team is looking forward to work together with you in doing some pilot studies. Finally, I would like to give you a look on our high throughput DNA extraction robot, the so-called oktopure. So the oktopure is an instrument really which was designed for a lot of samples in a short time period. Not only for plant breeders, this might be important, but also for service labs again, who are maybe getting a lot of newborn bovine samples and they have to genotype this in a very short period. So in total, this robot can extract 8 plates, 96 samples per run in parallel. So a lot of other instruments which are available can also extract, for example, 1 plate 96 samples during one run, and this one can extract up to 8. The whole extraction process is happening in this gray areas here, the M1. So M stands for magnet, and these are the so-called processing sites, meaning the entire extraction from the binding to the beads till the elution is happening here in this gray zones here. And the great thing of the oktopure is that it is really making an in parallel extraction, meaning in times, for example, when here we have an incubation time of 10 minutes, the robot is then extracting all the other plates and then starting again with the first plate. So we're really having a time efficient robot here, which is guaranteeing you by this a high throughput. And after 2.5 hours or 2 to 2.5 hours, you then have about 8 plates in total extracted. So meaning that you are having after 2 to 2.5 hours about 750 samples. The other positions on this deck here, what you can see is here the L1 position. L1 stands for lysis plate. So, for example, if you have your tissues or blood samples lysed in this 96 format, you simply spin down all non-soluble residues, and the oktopure then is transferring certain volume, for example 100 or 200 microliters, from this position here to this M1 position. And finally, we have also this yellow positions here, and these are the so-called elution positions, meaning here would be the ready-to-go DNA for your dose application. So the last step would be that the robot is processing the DNA, the purified DNA from the processing site to the elution site, and this line would be the ones that we would take out of the robot and use, for example, for genotyping and externation sequencing. You can see here also the buffer reservoirs, and here in the back you can see the tips which are stored here. So every processing site has one tip rack. For example, this M1 position would just use the T1 tips, and therefore you have also no cross-contaminations, because that's always something which is being discussed. So there are really no cross-contaminations possible, simply because we're using the same tip for the same extraction all the time. And you can see here also the tips for the buffers. So all the buffers are used, or you're using the same tip always for the buffer, and you have also cross-contaminations here. Just to summarize that, so the technology is a typical liquid handling machine. We have here magnetic rods which are under the deck and which are lifted up and down. The plate format are 96 samples, and the total throughput is 8 times 96 samples in parallel. So if you're going for an 8-hour shift and you're really working full speed without any cigarette break, then you will probably be able to extract about 3,200 samples per day. Again, that's a little bit depending on your shift and your time and so on, and also your protocol. Consumable costs are approximately 15 cents per extraction. Applications, again, you can use this DNA for all kind of things, and this robot is really optimized for the sbeadex chemistry in general, and especially also for the sbeadex livestock chemistry. Just one thing I would like to mention here is there's a certain capacity limitation in terms of blood samples. So the oktopure is just able to extract 50 microliters of blood as a maximum capacity. This is the only exception is chicken blood, because here we can dilute the blood. So this is simply because of viscosity and also technical reasons. So the limitation on the capability in terms of blood extractions on the oktopure is restricted to 50 microliters. On all other kind of platforms, you can even go higher or lower with the volume. That's not a problem at all. So, for example, the Kingfisher or Tican robot, you simply can go also for 200 microliters. That's not a problem. Then all other applications are no problem anyway. But please keep that in mind. So, to summarize this webinar, I told you this will be the 30-minute one. So, as a summary, again, one protocol for all livestock samples. The lysis is still offline, but all other sample types can be extracted in parallel in the 96 format on different kind of robotic platforms like the oktopure, like Kingfisher, Hamilton, Tecans, whatever, which is open. If you have a Kai Symphony, for example, then unfortunately it's not possible because this robot is definitely not an open platform, which can be supported by our chemistry. The DNA is really the DNA quality is really high, so you're really having high DNA quantities as well. And I've shown you that we have tested different kind of species. And finally, in the end, this is a summary. We are really offering this as a team, meaning sbeadex are the beads, oktopure is our unique robotic platform, and we are really offering you here some kind of very flexible DNA extraction solution, which is also offering you the possibility to reduce your costs for the consumables, which is always a very important issue. Other platforms definitely are needing consumables which are more expensive than ours. And what's always important, it's not just the flexibility, it's not just the cost, it's also that you have really the chance to achieve throughputs of 3,500 per 8 to 9 hour shift. So that's really something which you should keep in mind. It is a high throughput, but still very flexible DNA extraction solution. And by this, we're coming to the end. So I would like to thank you very much for your attention and also your time that you've joined the webinar in the afternoon. So if you have any questions, you're always welcome to ask me. You can find my email address here in this last slide. You can also contact me via phone, for example. So simply call the general office number in Berlin. And if you have any questions, I'm for sure open to answer all your questions. I will also send you the presentation after this webinar via mail in the next days. Again, thank you very much. Kind regards from sunny Berlin. So we have sunshine outside, and I would like to wish you really a nice day, and thank you for your attention. Bye-bye.